let us study about something called as a simple microscope. Okay, so we know just a lens, okay, single convex lens is used by the palm readers, okay, or we can use it to see small letters there, very tiny letters there. So that's a simple microscope. Let's see how it works. Okay, so that's the thing here. A simple convex lens. Okay, you just have a single convex lens here like this way. Now this is a principal axis. Okay, let me divide the convex lens in two parts like this way. Okay, now we know there is a focus here. This point is the focus. Okay, we must keep an object between the optic center, this is optic center over here, and the focus here. So let's keep an object here. Okay, so this is an object here. Now what happens is, first light ray is parallel to principal axis. Okay, after reflection or after refraction, it will bend and it will go like this way. It goes to the focus on this side. Okay, so this is the first light ray. The second light ray goes to the optic center like this. Okay, so you can see these two rays are divergent. Okay, they don't meet anywhere, but if you produce them like this, okay, produce it, okay, these two rays appears to meet here. They appear to meet at this point here. Here you construct an image. That will be the image here. This is the image. Okay, so an image is formed here. Now let's mark the parts here. Okay, so this is the object distance here. Okay, from here to here, the two object distance is u. This is a focal length. This focal length is represented by small f. Okay, and the image distance here. Okay, this is the image distance. That is usually we write image distance as v. Okay, in this case you're going to write as d here. Okay, why? Because we say d is the near point for normal eye. Okay, so that near point means what? If you read a book, okay, the minimum distance should be 25 centimeters. Less than 25 centimeters will cause eye strain. Okay, so the near point for a normal eye will be 25 centimeters. Okay, that will write here. So the near point for a normal eye, the distance d is equal to 25 centimeters. Okay, so we can see clearly at 25 centimeters. Less than 25 centimeters, it will not be very clear. Okay, and the far point, okay, if eye is normal means the far point will be infinity. Okay, same way, the far point for a normal eye, that will be is equal to infinity. Okay, this is a symbol for infinity. Okay, it's written like 8 sidewise. Okay, so the far point is infinity. Now we know the equation of magnification, that is m is equal to v by u. Okay, m is equal to v by u. Now you substitute these values here. Substitute v. Okay, v means what? The image distance, that is d here, but you also use a new sign convention, new Cartesian sign convention. What is it true? It states it like this way. Okay, distance on the left side is minus, right side distance is plus, height upwards is plus, height downwards is minus. Now if you see all these terms, the distance come on the left side, so you write as minus. So instead of writing v, you write as minus d. V is equal to minus d. And of the distance u is coming on the left side, so write as minus u. Okay, so we're not writing plus u is equal to minus u. We're saying instead of writing of the distance u, you write as minus u. Okay, that's the thing here. Okay, so now we substitute here. So what we get is m is equal to minus d by minus u. Okay, right now. So plus sign, minus and minus cancel, so we get here plus terms. So m is equal to d by u. This is an equation for magnification produced by the simple microscope. Okay, a simple microscope is just a single convex lens only. Okay, sometimes in the lab used for biology lab used for dissection microscope also. The dissection microscope you just have only a single lens. Okay, so this is the thing here. So m is equal to d by u. D is the near point for a normal eye, that is 25 centimeters. Okay, u is the object distance. Okay, so this is the thing here. Remember, the object should be kept between the optic center and the focus. Only then you will get the virtual image like that way. Now there's another way of deriving this equation. Okay, let me see that and write it down. Okay, I'll erase the board now. Okay, now let's see another method to find the magnification produced by the lens. Okay, so what we do is we substitute the lens equation. The lens equation is what? That is 1 by v minus 1 by u is equal to 1 by f. Okay, in this equation for magnification, that is m is equal to v by u. When you do this and solve it, we get the equation here. Okay, we get magnification m is equal to f minus v by f. This is an equation we get here. Okay, so now what we're doing is, so we'll get this equation. Now so you can take the denominator separately. That will be, this will be f by f minus v by f. So what happens, f and f cancel, so we get here 1 minus v by f. Okay, in this equation we get here. Now what we do is, in this equation substitute here v. Okay, instead of v, what we have, that v means image distance. We, instead of v, we write as d here, that to minus d. Okay, using sign convention, left side distance is minus, so minus d here. So what happens is 1 minus, instead of v you write as minus d, so write as minus d by f. Okay, so minus into minus will become as plus, so 1 plus d by f. This is the equation for magnification. Okay, this is another equation we write here. Okay, now we have two equations for magnification. One was we got here m is equal to d by u. Another one is we have here 1 plus d by f. Okay, so here d means what? The minimum distance for clearing, seeing things clearly, that is m, or you can say near point for a normal eye, that is 25 centimeters. D, f is the focal length of the lens. Okay, so it's always, it will be larger. Okay, the image is larger in size. Okay, so this is used in a simple microscope. Thank you for watching.